slipped a little bit, partly for my fault. And I'm here representing Owkin. We're a data science company in France. Our objective is to do precision medicine, and our basic trick is using federated multimodal patient data to pull that off. And let me find the little click box here. So I like to start off my presentations usually with a slide to explain why precision medicine is important. And this is actually a slide that I pinched from Dan von Hoff in 2016, where he basically made this very disturbing picture. So this picture here is showing a glimpse at all of the clinical trials that happened in 2016 as represented by clinicaltrials.gov. And what you see is that they're broken out through all the different kinds of cancers that are out there. So these are all the trials that involved PD-1 or PD-L1 treatments. So they're the best oncology drugs that are out there these days today. And it's broken out across the different kinds of cancers. And in the outer layer, you see phase 1 trials, which involve 30-odd patients. You have phase 2 trials in the middle, which are several hundred, and phase 3 trials in the very center, which are thousands of patients. And what's interesting is that there are dots everywhere. Each dot represents a trial. The number of dots has nothing to do with the response rates. It has everything to do with how many patients are available in each indication. And what's also interesting is that the response rates in all of those boxes are less than 50%, and in some of them less than 20%. And so what this is telling you is that there are thousands upon thousands of patients who are getting treated when we know that they're most likely not going to respond. And this is just an appalling situation that happens throughout healthcare. And it really comes from the fact that we just don't understand the biology of disease well enough to send patients to the right treatment. So the solution to this problem is precision medicine. And precision medicine is an idea that's been around for a while. MD Anderson has been a big proponent of this, where the idea is to basically look at all of the confused biologies that might be in a particular kind of cancer, figure them out, and then find drugs for each subtype that exists within that, and then you can actually treat patients with the right drug, and at that point you can expect much higher response rates. So our approach at Owkin to doing this is to essentially use multimodal data about patients to get as much information as we can about how patients differ from each other. So we like to work with clinical, imaging, and molecular profiling data. We put it all together, and we use that to try to take a complex cancer, something like triple-negative breast cancer, and break it out into subgroups where each of the patients have different biology. The reason why this matters is because if you look at TNBC, that's triple-negative breast cancer, the outcomes in that for that disease are terrible. Patients get it, and basically we don't have any good drugs to treat them. And the main reason for it isn't that there aren't drugs that might treat TNBC patients. The main reason is that the diagnosis is too approximate. It lumps together many very different biologies, which means that if you're looking for a target in TNBC, it's really hard to find any signal because it's all jumbled together. And if you do find a target that works in TNBC, only 10% of your patients, at best, have the right biology. So when you run a clinical trial, it fails. So you can't get drugs out on the market to treat it. So the right solution is, in fact, to break it out into subgroups where you have different biologies, which then allows you to do two things. First of all, you can find targets that work for a subtype. And second of all, you can try to find biomarkers that will help you steer patients to that right drug treatment. And that allows us to then do what we really want to do, which is matching the patients to the right drug. Oh, did it not click? There we go. So this slide here is sort of a one-slide overview of everything that Owkin does to pull off that mission. And on the left, we have a bit of history, and on the right, we have what we do. So the bit of history is we've been around since 2016. We've grown a lot. We're based in France mostly, but we also have a lot of presence in the UK and in the US. We've cut some very nice big deals and raised a lot of money. On the deal side, we made a very nice deal with Sanofi a couple of years ago, where they made a big investment in our oncology exercise. And BMS made a big push into cardiovascular for us. What we do is summarized by what's on the right-hand side. And basically what we do is we take the multimodal patient data and put that into our platform. And our platform basically has three components to it. The first is federated data access, which allows us to reach into those multimodal patient data in a safe manner. The second is multimodal integration, which allows us to connect data elements across many different types. And the third is AI, which essentially we use to generate the outputs that come out of our platform. The outputs fall into three big categories that sort of represent the business history of the company. So in the top is diagnostics. So in the early days, we were very much like a number of companies that are out there, like Page AI or Path AI, where what they're doing is essentially looking at patient data to predict disease outcomes. And that's a way to sort of send patients towards the right kind of treatment. The second the output that we have is what we call pharma solutions. And really what this is is trial optimization tools, things that allow you to sort of pick out patients more effectively or compare them to outside populations or even analyze your signals coming out more cleanly. And that actually is something of great interest to pharma companies because they're trying to improve the efficiency of their operations. So that's led to a lot of revenue generation for us, which has been extremely helpful. And then the third is what we've always viewed as our future, which is drug discovery. So our real objective is to try to identify new disease subtypes in diseases like triple-negative breast cancer and discover the biologies that can be used to treat those new subtypes, and then maybe find some new drugs that we can put onto the market, which will enable the treatment of patients that previously couldn't be addressed. So the later slides now just go through the different pieces of what's on that outline slide. So first of all, the patient network. So what we've done over the last 5 years is built up a large network of healthcare systems that are actually working with us to connect together their patient data. We have a number of clinics, especially in Europe, and that really is for two reasons. It comes from, first of all, the company started in France, so we started building our network first in France. But because we were building our network out in Europe, which lives under GDPR, there were very strict rules on patient data privacy, and that's forced us into getting good at connecting to data in a very safe way, which has turned out to be an enormous benefit. So, which is also even attractive to North American centers who are now expanding quickly in our network. So the next slide explains our approach of data federation. So if you're going to put together information from different sources, from different healthcare systems, for example, the traditional thing to do is data pooling, where you might make a copy of the data from each clinic, put it into a protected central space that you administer, and then that's where you do your model training to understand what's going on in a population. Federation does it exactly the other way around. So you leave the data with the institute locked away behind their security firewall. You send the model behind that firewall to learn what it needs to learn about that clinic's population, and then the model travels between all of the institutes to basically learn what's the overall picture across all of the populations. What's interesting about this approach is that, first of all, it's completely secure and private because we never directly see the patient data. It's controlled and administered by the host institution. What's really attractive, even outside of Europe, where GDPR isn't reigning, is that the institute controls and owns the data throughout the whole process. And this is very attractive for North American institutes as well. And finally, it's efficient and scalable. So many of you know that Google has been using federated approaches to do cell phone analytics for years. And the reason why they do it is for two reasons. First of all, it simply is much more efficient. Moving models around is a lot easier than moving data around, and so you can scale it much more readily. And secondly, if they were to centralize everyone's personal cell phone data, they'd be in court until kingdom come. So they obviously don't want to do that. So on the right hand are three examples of where we brought a federated approach to the world. The first is Melody, where we essentially worked with 10 pharma companies to connect their drug screening data together. So this is like the most precious information that pharma companies collect over time. It basically says how well compounds with secret structures that they developed bind against proteins using assays that they've developed, and they just don't normally share this with anyone. However, these companies wanted to put their binding data in the context of other people's binding data, and so they adopted a federated model administered by us to basically do a larger study. And I'll tell you a little bit more about that in a later slide. The next example is where we worked with hospitals in Europe, both in a smaller collaboration called Health Chain, and then later on in Optima, going across the entire continent to connect clinical information together in a similar mode. And the third example is the most recent. It's the Bridge to AI collaboration, where essentially we worked with a couple of universities to connect voice data that might be informative on neurologic conditions. And we're beginning to look into how we might analyze those data as well. How the federated learning platform works is basically that we have the compute happens locally at each of the data centers. We send the models out at the same time to all of the centers, learn what's happening locally, and then we iteratively bring it back to the aggregating server where it's averaged to essentially reduce the information about where data came from. And we cycle back and forth until you essentially have the information from all of them combined in an essential fashion. On the right-hand side sort of walks through the different components of what our Substra platform does. The Substra is our federated learning platform that's available, and how it solves the different things that are needed for federated learning. One thing that's worth noting is that we've made the Substra software publicly available. It's on GitHub. You can load it down anytime, and it's hosted by the Linux AI and Data Foundation. This is just a little bit of detail on the Melody collaboration. So when you look at the recording, you can go into the slide in detail, but I'll just tell you a few points about it. The main thing to note is that the pharma companies that are involved are the largest pharma companies in the world. So these are sensitive companies that were willing to pool their data effectively to do this kind of analysis because Federation made it a safe thing to do. The results on the right sort of show how much improvement they were able to get coming out of it. And it's fair to say that we got moderate improvement on binding prediction if you're asking a very narrow question, which is how well does this model, does the collective model predict what the binding that I have between my compounds and my assays. Where it does give you significant improvement, however, is in the applicable domain. If you look at the chemistry and the assay domain covered by the larger set, it's quite a bit larger than what you have on the individual sets. And this is despite the fact that most pharma companies are typically testing the same kinds of things with the same kinds of assays. What we generally do with data once we connect them together, especially in the clinical domain, is we build models. And this slide here gives you an idea of the kinds of models that we've been building over time. So the example on the left is actually a particularly clever model, which is called AG2RNA, and it's so called because what it does is it predicts the expression of about 200 genes at different locations across a tumor. And it does that entirely looking at one source of data, which is the standard routine H&E pathology image that every patient gets in modern cancer care. How does it do this magic, you might ask? Well, it does it because what we had, we trained the model on a set of data where we had thousands of patients' images, H&E images, paired with RNA, with bulk RNA-seq, so gene expression measurements from each tumor. What's interesting is that the RNA-seq data, which tells you about how much each gene is expressed, doesn't tell you anything about the spatial distribution. But the image features that associate with the expression of each gene are distributed differently. So how cells group or how they're shaped is different from one location to another. So you end up with very strong spatial signals for each of the genes, and you can essentially infer from it the spatial distribution of about 200 genes across a tumor. That assay, getting the spatial distribution of a bunch of genes across a tumor, is very expensive, it's non-standard, and it's almost never done, except in very well-funded pharma trials. So essentially, what it allows us to do is predict something really hard to measure from routine data that you already have in routine care. We have a whole bunch of models that we've generated, and the examples on the right are some of them. And all of these models share that common quality in common, which is that the idea is to predict something hard from something routine that you already have. We have 2 relapse risk and MS Intuit that are proceeding towards approval right now in the European market, and we're moving them towards approval in the US as well, which essentially predict either disease outcome or they predict drug response. And we have others that are going after genomic state, very much like what the AG2RNA model has. What's behind those models is our AI engines. And our AI engines basically are a whole collection of AI tools that we put together to solve different problems. Here they're grouped into 3 big categories. The first category is the target discovery engine on the bottom right. And basically, this is tools that allow us to first of all, find the subtypes that might exist within a group of patients, and then within each subtype, discover targets that stand out as being typical for that subtype, and then we can prioritize them based on how druggable they are or how causal they might be or other things that we might infer from the information that's available. The drug positioning engine on the bottom left is exactly the same tool, but it runs the other way around. If you come in with a compound that has known targets, you can use that to figure out which subtypes might be most applicable to that compound. And finally, we have the drug optimization engine, which is the bread and butter for most of our pharma collaborations, which basically allows you to accelerate development using tools that either select patients or make comparator arms out of, in this case, federated populations in our network, or essentially, or to clarify outcome signals using analytics like covariate adjustment. We also are working on a very exciting consortium right now as a new source of data that is going to give us a unique look into the area of tumor immunology. So basically, our goal was driven, this project was driven by a desire to build an atlas, to essentially build the definitive immuno-oncology discovery resource out of spatial omic data. What is spatial omic data? Spatial omic data is basically it's new technologies that allow you to directly measure the kinds of things that HE2RNA predicted. The expression of thousands of genes in different locations across a tumor. Why is that useful? Well, it's useful because the tumor and the immune cell, the tumor cells and the immune cells in a cancer patient actually interact on a very local level. What typically happens is a bunch of immune cells nudge up to a cancer cell and decide whether to kill it. And the whole interesting interaction is how that happens, and that all happens quite locally in particular parts of the tumor. What that means is that if you're trying to interpret the mechanisms involved using the traditional approach of grinding up a tumor and measuring gene expression, you will typically lose most of the interesting signal because it's drowned out by everything else that's happening that has nothing to do with that interaction at that location. What spatial omics allows you to do is basically chop up the tumor into lots of little bits, find which ones have interesting tumor-immune interactions, and then zoom in on those at the mechanistic level. And that's the power of it. That's the wonderful thing about it. The annoying thing about those assays is that they're very slow, they're very fussy, and they're extremely expensive. So as a result, the biggest studies that anyone's run so far involve less than 30 patients. And this, to us, looked like an opportunity for an industrial enterprise like us to make a significant impact. And the approach that we're doing is to start up a new consortium involving a number of medical centers in our network, where the goal is to take 7 cancer indications, collect about 1,000 patients worth of data in each of those indications, using the spatial omics technologies provided by NanoString, the GeoMX and the CosMX platform. The idea basically is that the medical centers are providing the patient samples. They'll be doing the data generation on their premises. The NanoString is going to provide us at heavily discounted rates the equipment and the reagents to do it. We'll be coordinating the data integration. We're funding the whole thing. And together, we're all going to be discovering new cancer subtypes, biomarkers, and targets specific to immuno-oncology. And the data generation is going to happen over a few years. And we already have pilot studies underway to get ourselves familiar with these new data types and how to make sense of them. All of this is part of our general trajectory as a company, where we'd like to evolve from what we used to be to what we want to become in the future. So remember, what we used to be was primarily a diagnostics company. And what we really want to be is much more of a drug discovery company. And it's helpful to sort of put us in the context of other companies that are generally perceived to be in our space. So particularly in my previous life, when I was in pharma companies, there was an awful lot of confusion about what AI companies are. If you look at all the symbols of the companies present here, you would typically find in a conversation that half of the audience, if not more, were confused about what the difference was between Recursion and Path AI. What is the difference between all these different companies? So what I find is helpful, and helpful for describing our trajectory, is to think about 2 big parameters that separate different companies in this space. Parameter 1 is a very obvious one. What does the company sell? So if they sell models, like we did in the past, they belong in the left-hand category. If they sell data, then like what we're sort of drifting through indirectly with our federated network, then it's like the companies in the middle. And the right-hand category are the ones that we want to join, where essentially what they are selling is a drug pipeline, typically purchased for later development by pharma companies. Now, all of those companies are quite different from each other as well. So to understand that space, it's helpful to then break it out by the other dimension, which I like to focus on, which is what does a company do? What are its core capabilities? So if you're going to be a drug discovery company based on AI, you have to do certain things. The first thing you have to do is have some data to do your AI on. After that, you'll need some way of discovering targets, typically, then a way of validating the targets that you found, and ultimately trying to match some compound to the targets that you validated. And companies that have entered into the space of AI biopharma have done this in different ways. In the early days, the ones on the bottom basically moved into the field by getting really good at matching a compound to a known target. So someone would come to them saying, here's a new target that I'd like to address. They would find a way to get a target that binds to it properly, to get a molecule that binds to it properly, and then they could develop that and build their pipeline on it. The later entrants emerged next, where what they got good at was mining the public literature and public data resources to find new targets, to find targets that can be addressed, and then to match molecules to the targets that they discovered. In situ and Recursion then opened a new plane where their idea was to actually use complex phenotypic assays to discover new targets that aren't even being written about, and thereby open up new opportunities for targeting in that space. And then the top section is the one that we think we'll fit in, where what really defines those companies are the ones that have large collections of patient data. And what that allows you to do is the kind of subtyping that I described, where you can essentially create new markets within new indications that you can then look for targets in. So one thing that people often ask me is, well, if we're going to be going down the bandwagon of drug discovery, what happens to the diagnostic and pharma solutions? Are they still part of the game? And the answer is absolutely yes. So our goal, remember, is to try to find the right drug for every patient. our drug discovery connects very directly to that. We're going to be essentially growing our AI-driven in silico discovery methods to do that. We're going to be building out experimental validation capabilities, and we'll be doing a lot of partnering to pull in molecules and optimize them. So that seems very obvious. The pharma solutions are still really helpful because as we start moving towards clinical trials, we will want to run our own, to optimize our own clinical trials, and that will give us an advantage when we're validating them in the clinic. The reality also is that we will be running pharma solutions deals with pharma companies all the time, because that source of revenue allows us to stay cash flow positive to this day, even as we're growing, and that is extremely helpful for our financial health. And then finally, the diagnostics are actually the core of our business and will remain the core, because ultimately many of the drugs that we find are going to be addressing subsets of a patient population. You will need biomarkers to identify which patients belong in that subset, and so we'll be needing that as a core part of what we do every time. So this final slide that I have here just sort of outlines how why we think Owkin is going to make a difference on the market. And it really comes from the fact that we start from patients' data, we use that to advance precision medicine, which benefits patients, and that makes more and more clinics more willing to participate in our network and help us in this overall operation. How we do this is with the stuff in the middle. All the tricks that I described in the previous slides essentially build out towards how we're able to do this. And the bottom line is probably one of our most defining characteristics, which is we've had incredibly strong finances. So last year, we raised about $300 million worth of cash. We haven't yet spent it because we've been cash flow positive through this whole period, which means that we are in a very strong position to make bold moves into generating new capabilities and growing towards our new future. One last slide I wanted to add. So the conference organizers, the workshop organizers wanted me to talk a little bit about how we would work with Google Cloud. And basically it falls into sort of three big categories. One category is by federating partner data sets. So essentially, we have a core strength in federating data in a very secure fashion. And this is something we think fits in very well with Google's larger mission of trying to connect data resources from different places. There's also a ton of research and development work that we could do together. Google is great at some things that we'll never be good at, and we're good at a few things that Google can actually make good use of, particularly in the life sciences space. So we think that there are definitely R&D projects that we could begin that would make an awful lot of sense. I want to draw particular attention to federating biomedical data consortia. So if you look at the Broad Terra initiative, or if you look at CancerLink, or if you look at other big consortia between different clinics, one of the biggest problems that they're running into is that there are data silos. Institute number one can't or doesn't want to share their data with institute number two. Pharma companies never want to share their data with anyone. And it fundamentally comes from two concerns: commercial interests or privacy constraints. Both of those can be addressed very nicely by using a federated approach, as was shown by both the Melody and by our clinical networks. So I think we should be extending this as much as possible to many different diseases. And we would be happy to help Google take a leading role in extending this kind of data sharing in as many ways as possible. Obviously, we'll be pursuing our own commercial aims for some of them, but for most of them, if you think about neurological sciences, we don't currently have a plan to operate in that space. So we would love to help that happen, and we sort of don't care who does it, as long as it's done well. And so finally, I just wanted to end with the summary slide and take any questions that you might have. So each site can be thought of as a batch of samples that are coming in that might have differences between them. Those do happen, and essentially we have to try to build models that can account for that. So the way we do that is when we're putting a network together, we try to make sure that we have different imaging technologies represented at each site, so that then you can essentially try to understand what the differences are per site, and then try to understand the trends when you go across sites. The bookkeeping of all that of how all that works is deeply within our data science team, and happily not my responsibility, so I wouldn't be able to explain it to you clearly. But like any good AI team, they're thinking about